All right, good morning, everyone. We're going to go ahead and get started. It's wonderful to see a packed room and to see so many smiling faces so bright and early in the morning. I just wanted to share some opening words, and then we're going to get underway with our program. So I recently listened to a series of podcast episodes on pandemic preparedness, as one does. These were produced by the 80,000 Hours podcast, which I highly recommend. And it was really surreal to listen to experts talking before March 2020 about the best way to prevent, detect, and respond to emerging pandemic threats. And there were a lot of excellent ideas shared, things like pathogen agnostic clinical testing, running through tabletop pandemic preparedness exercises, being more transparent about lab accidents. But not once in 9 hours of interviews did I hear anybody mention wastewater surveillance, which, well, and I should make the caveat that there was mention of environmental surveillance, but that was in the context of air sampling, not water or wastewater sampling. That just tells me that a short 54 months ago, we really weren't having a coordinated national discussion about using wastewater monitoring for public health. Fast forward to 2024, and I think it's fair to say that that has changed. This year was a landmark year in terms of workshops, conference sessions, trainings, meetups, and other events focused partially or wholly on wastewater surveillance. And one of my favorites was the wastewater workshop at the Council of State and Territorial Epidemiologists National Conference in Pittsburgh just in June, because it was remarkable to be in a room full of professionals, many of whom identified as wastewater epidemiologists. That's a job description that didn't exist before 2020. So I think it's fair to say we are definitely talking about wastewater now. And the Wastewater Disease Surveillance Summit is going to give you 2 more days of conversation on the topic. So over the next 2 days, you're going to hear from data scientists, epidemiologists, laboratorians, researchers, utility professionals, and others about the whole life cycle of wastewater surveillance. You're going to hear a lot of information on the topics that you're familiar with, but also new targets that you might not be so familiar with. You're going to get a chance to explore 30 fantastic poster presentations, connect with your peers to talk about challenges related to selecting and onboarding new targets, and just generally get to hang out and have fun. This summit is made possible through funding to WEF from CDC under a cooperative agreement. So we're incredibly grateful to CDC for making this summit possible, but we just want to note that the opinions expressed by WEF staff, by summit presenters, summit participants do not necessarily represent the official position of CDC. And then I realize I need my clicker. I gotta go to the next slide. The wonderful program that you get to experience over the next 2 days was put together by our intrepid program committee. So Annabelle, Ariel Christensen, Megan Fuke, Martha Johnson, Nathan Lacrosse, Brenna Magruder, Aaron Morin, Naoko Munakata, Smiti Nepal, John Person, Steve Rohde, Rekha Singh, Eric Vaughn, and Jeff Wenzel. They have on their name tags that will say program committee. If you meet one of them, please say thank you to them. But yeah, they had a lot of wonderful ideas, energy, and a lot of tolerance for the many, many emails and meeting invitations that they received from me and Jenny over the past 8 months. Okay, a few housekeeping items. There is free Wi-Fi. There is also, please help yourself to coffee, tea, and soft drinks throughout the venue. And there are masks available at the registration desk if you would like to use one. And then we have a few asks of you. So when you came in and picked up your name tag, if you did not get a chance to sign in the old-fashioned way with a pen and paper, please make sure to do so. But we are trying to minimize our use of paper, so we did not print out a program this year. Instead, we have a digital agenda, which has pretty much everything you'd want. It's got sort of an agenda at a glance. You can also click on that. There are buttons in there to click on to get the full agenda with full speaker names and titles of presentations. There's information on safety and security at this venue, ideas about where you can walk to get lunch, just about everything else in there. If there's something that you're not seeing in the digital agenda, or if you have a question, comment, or concern, please seek out one of us. We are your facilitators. So Jenny and Carly and me, we're available to answer any questions you might have. Okay, so now I have to apologize. So we did not do a very good job anticipating how many of you were going to want to travel to Atlanta in August to talk about wastewater, which is why there are some of you who are in the main room and some of you sitting in overflow rooms. It was the best solution we could come up with, and I'm really grateful to the event center and Barbara in particular about kind of brainstorming how to make this happen. Which is all to say that we really request that you take a minute to spend time in the overflow rooms for some of the sessions and rotate through. If you all are sitting in the main room now, maybe after the break, go into one of the overflow rooms to give somebody else a chance to come into here. In the overflow rooms, though, as the folks who are in there now know, you will be seeing the presentations, the panel discussions. You will be able to ask questions as well. So you really will be part of the conversation. And then the last request of you is to consider joining us for a meetup right after the programming concludes today. It's right across the street. There's a link with a Google Maps, directions in your digital program, but we're going to meet up at Politan Row at Colony Square around 5:30. Just informal. Dinner is on your own, but we'd love to, kind of continue chatting with you all. Before I turn it over to our keynote speaker, I wanted to go ahead and ask you 2 poll questions. So we'll see if this works. The first question is, what US state or country, if outside the US, do you work in? And you can answer more than once. You might work in more than one state, more than one country. So we'll give you all a minute to answer this. No repeats so far. Oh. Okay, makes sense that Georgia is winning at this point, given where we're located. We'll wait just another minute or two. And we've got France, space. I like that. Guam, awesome. Texas, this is great. Okay. I'm going to go ahead and go on to the next poll question. So which sector best describes the one you work in now? So academia, federal government, nonprofit, private sector, public health laboratory, and wastewater is down, wastewater utility. And then there's an other option if none of those capture where you work. Okay, so about a third from a health department, whether that's state, local, territorial, or tribal, about a fifth from academia. We've got a good representation from federal government, public health laboratory, private sector, and wastewater utilities bringing up the rear. Okay, awesome. Thank you all. I'm glad that worked. Now I'm going to turn it over to Arielle Christensen to introduce our keynote speaker.
Good morning, everyone. I'm extremely honored to be here to introduce our keynote speaker. Kody Kinsley serves as North Carolina's Secretary of Health and Human Services, overseeing our department with over 18,000 staff and a $38 million budget. With experience centered on health policy and operations, Kinsley worked on digital healthcare transformation, national education and labor policies, and served as the COO and CFO of the U.S. Treasury. Secretary Kinsley's 3 priorities for our department include investing in behavioral health and resilience, improving child and family well-being, and building a strong, inclusive workforce. Under his leadership, North Carolina expanded Medicaid and received the largest investment to bolster mental health system in over a decade. We're extremely lucky to have him in North Carolina. Kinsley grew up in Wilmington, earning his bachelor's degree from Brevard College and his master's in public policy from the University of California at Berkeley. So I'm extremely honored to introduce Secretary Kinsley.
Well, thank you so much for that kind introduction. And I really regret that I can't be there with everyone in person. And I want to first say I have to update my bio to add wastewater to one of my top priorities because it is something that I have been very excited about. And so when the team asked me to, or invited me to be a part of this conversation, I was happy to turn down every other thing that I have to do Monday mornings at 8:30 to be here. So I really appreciate the opportunity to join you all. Our wastewater monitoring efforts are incredibly important to the Department of Health and Human Services, and of course, important to our entire country. We're excited of our launch in 2020. And as we look back, and the opening remarks alluded to this, the COVID pandemic was a huge challenge to all of us in many ways, including to our public health system. And people are rightly asking today, and it's perhaps the question that I get most often, are we indeed better prepared now coming out of that response than we were going into it? And I believe without question that we are. And I believe it is because in large part, we've made investments in North Carolina and across this country in systemic changes and knowledge that we can help parlay into the future. One of those key advancements that we made was investing in wastewater surveillance. And in just a few short years, this has become an important tool that helps every community understand the health of their community and complements other health surveillance tools that you all know well. We learned during COVID-19 when we started pushing this data out onto our public dashboards that people really love poop data. And my team has grown tired of my love of poop jokes, but I think it's important to stay in the gutter sometimes. Seriously, wastewater surveillance is an important detection tool for pathogens because, as you all know, they can pick up asymptomatic folks and they're passive. But I think, and this is a really important aspect of wastewater surveillance, is it's something that the public can grasp. It's something that they understand innately from a young childhood. They understand the toilet, they understand the process, whether they understand exactly how the wastewater system works, fine, but they get that it's all going somewhere and that we can potentially test it. And then they can see, because of the wastewater watersheds, where we're seeing increased viral load of various things that we're testing for, and they can connect that, oh my gosh, my kid's school is in that district, and we're seeing a tick up, and that's going to be caught a little bit earlier than we're going to see other indicators get caught. This allows us to have that critical public conversation that we know is foundational to our COVID response and is really foundational to public health. As I like to say, the first word of public health is public. And thinking about how we communicate about public health and how we work with folks is foundational. Now, we all know in the room and virtually that wastewater surveillance is far more complex than the ease at which the public can grasp it. And it requires immense collaboration with researchers, community members, local health departments, and others around sample collection and analysis. It requires being transparent about the data and developing visualizations with data scientists to make sure that folks can really understand and grasp what's happening and that we can do that in a fair and transparent way without creating unnecessary uncertainty. And it also requires us to be really transparent about the data that we can get from that, the information we can get from that, and how it drives our decisions, because just because it's ticking up doesn't necessarily mean that we're going to do a certain thing differently or when we're going to do a certain thing differently. And helping folks understand those levers, we know that it can be crude in some ways, and so we have to be able to couch that appropriately. Most of our public health system and our public health surveillance data that people are used to relies on healthcare services. But again, with wastewater, we're able to get a glimpse of what populations are doing and experiencing when they're not seeking care. That's been particularly important for us in North Carolina, as a state that just recently expanded Medicaid. And by the way, we have nearly 550,000 people now enrolled in Medicaid expansion, which we're very proud of that work and the speed of which we've been able to get enrolled. We knew that going into COVID, we had a legacy of nearly 1.2 million people, 10% of our population, that did not have health insurance, which meant that they interacted with the health system in a very different way. And we're still breaking down those behavior patterns and modifying them with access to insurance, but it meant that our normal surveillance methods that really ride the backbone of the healthcare system wouldn't be able to capture those people in certain ways. We also know that this virus preyed on people like other viruses with chronic diseases who are vulnerable, living in communities that are left behind in many ways. And those systemic barriers really compound with lack of health insurance. And so we had a double gap in our knowledge of these historically marginalized populations and the impact thereof. Wastewater surveillance allowed us to really get a view into that health disparity and that health equity gap in a way that is really critical for us to be able to shape our response and to be able to meet people where they are truly. We are so excited that our department has been selected as a new center of excellence for wastewater surveillance. I'm so proud of our team and all of the work that they have done to really lift this out of the ground and out of nothing. In 2020, we were one of the first states to pilot wastewater surveillance, and we were grateful for funding from our state legislature, from the North Carolina Policy Collaboratory, and our close relationship with the University of North Carolina at Chapel Hill to make that possible. Again, as I spoke of earlier, these collaborations, the behind the scenes on wastewater surveillance is far more complex in contrast to the elegance at which the public can truly understand. I am so bullish about wastewater surveillance, and I hope you can hear that in my voice. I think that there's dramatic potential for testing for influenza A, H5N1, even potentially STIs or GI illnesses. I'm so concerned about the uptick in syphilis that turns into congenital syphilis issues for families and for newborns, and a number of other complex communicable disease issues, and our availability of wastewater surveillance is another tool to identify and define and to target our resources is going to be really critical and something we can parlay this into our broader public health mission. I know that over the next 2 days, you're going to have a lot of hard discussions, and I invite you all to really tune in, to take your phones, throw them away, and stay plugged into this conversation. You rarely get the chance to be in the room with each other, to hear from folks. You all are pioneers. You're pushing forward in a space that just a couple of years ago was pretty much unheard of by the public and not grasped nearly as broadly as we have been able to use this crisitunity to parlay into. And so I invite you to really bring your entire selves into the conversation, to use your brain power to push this work forward in an amazing way. Let's continue to build a sustainable wastewater surveillance strategy for the future together. Again, I can't emphasize enough the dramatic opportunity and elegance of wastewater surveillance. It is passive. It does not put more work on the healthcare system, and everyone poops. So thank you so much. I appreciate your time, and I hope you have a great session together.
Thank you so much, Secretary Kinsley. All right, I think we're ready to go. I'm going to call up Smiti Nepal as our moderator for our first session. And just to introduce Smiti, Smiti joined EPA's Office of Wastewater Management in 2016 with a focus on water reuse, nutrient removal and recovery technologies, wastewater surveillance, and emerging contaminants. Before EPA, Smiti worked with the Maryland Department of the Environment on solid waste landfill design review and permitting. She received her Bachelor of Science in Environmental Engineering and Master of Science in Systems and Ecological Engineering from the University of Florida. She also has her Professional Engineering license from the state of Maryland. Welcome, Smiti.
All right. Thank you, Anna, for that kind introduction. I'm incredibly excited for the next couple of days of presentations and discussions. We have so many interesting topics and ever-growing utilization of wastewater surveillance. It truly is amazing to see how far we have gotten in 4 years. And this is the credit to you all and all your efforts. So thank you, everyone. So for this particular session, you will hear from our presenters on diverse work that they're currently doing when it comes to subsewershed sampling. And the utility of this type of sampling is very unique and has a direct impact on decision-making for both environment and the public health. So today we have talks that talk about sampling strategies that can be scaled up and down depending on needs, and some really focusing on data and practical consideration for sampling upstream that have an impact on communication and looking into different targets. And really, the other unique aspect of this is looking into utilities' perspective on dual benefits of upstream sampling. And this really highlights how we can leverage sampling to meet both water quality and public health information. And last but not least, we have information on aircraft and airport sampling as well. We are going to have some extra time at the end of the presentation, so we'll be taking questions at the end. So without further ado, I would like to introduce you to our first speaker. So Dr. Lauren Stadler, who is an associate professor of civil and environmental engineering at Rice University. She earned her bachelor's in science and engineering from Swarthmore College and her master's and PhD in environmental engineering from the University of Michigan. Her research focuses on wastewater-based epidemiology, environmental antibiotic resistant, wastewater and resource recovery, and environmental synthetic biology. She has been collaborating with the Houston Health Department and Houston Public Works since the start of the COVID-19 pandemic to develop methods, implement and operationalize a citywide wastewater monitoring system that informs public health action. Dr. Stadler?
Okay, thanks so much for the introduction, and so excited to be here and to see so many of you in person. This is such a great event. So this is a session that's focused on upstream sampling, and we tried to cover kind of a diverse suite of applications. So today I'm going to talk about our pre-K through 12 school surveillance and how we've really tried to focus on actionable surveillance and vaccine-preventable diseases. So of course, I want to acknowledge this is part of a huge effort that is collaborative between the Houston Health Department, Houston Public Works, and Rice University. So many people have contributed to this work, and not even all of them are listed here, are named here, but this has been a huge effort that really started kind of in early 2020. And our success is largely due to this very successful collaboration between our groups. So just to give some context of the Houston system, which I feel like we've talked about quite a bit, but it's a big city. We have many different wastewater treatment plants. We're kind of a unique city in that we have 38 wastewater treatment plants. And in our monitoring system, we collect samples from about 115 sites every week. 38 of those are those wastewater treatment plants, which on this map are showed as shaded areas. But actually, the majority of our sampling sites are facility level. So they're collected from manholes located outside of facilities. And the majority of our facility samples are public schools. So we have about 46 public schools that we currently collect samples from. And all of these sites, samples are collected once a week. So the motivation that really kind of got this school program started was early on in 2020 because we know respiratory viruses, this is really before we knew a lot about COVID, we knew respiratory viruses are a leading cause of hospitalizations in children. And we didn't really know that much about COVID in kids at that time. And we know that previous work had shown that schools can actually be sources of respiratory viral outbreaks within communities. That motivated the interest in implementing some school level surveillance using wastewater. And then the Houston Health Department already had a lot of existing ties with the public schools and public school system. And so they wanted to use that existing relationship and really invest in it and use it as a springboard to encourage vaccination and outreach within communities. And so there's a clear kind of line for actionability from this data. This is a map of the schools that are monitored within the Houston system. As I mentioned, there's 46 schools. This is the largest school district in Texas. We have 3 different clusters across the city, a north one, a southeast, and a southwest one. Samples are collected from these once per week, and there's basically 3 different routes that are driven to pick up these samples. We started this sampling in December of 2020 and initially just started monitoring for SARS-CoV-2, and we've expanded the list of targets, which I'll talk a little bit more about. And these schools were originally selected because they were located in areas of Houston that had the highest burden of COVID-19 in November of 2020. And so we now have over 3 years of data on SARS-CoV-2 wastewater surveillance at these schools. Samples are collected using auto samplers. So the health department had the foresight to really invest really heavily and bought a ton of auto samplers really early on in the pandemic when they were becoming really scarce, and invested and put these in these manholes. So they're all located in these manholes, either inside or adjacent to manholes that only receive wastewater from those schools. They're programmed to collect samples every 15 minutes over the school day or approximate school day, 6 to noon. And then these samples are collected once per week and brought to our labs for analysis. The Houston Public Works helped with the installation and location, by figuring out the location for all these samplers, but it's Houston Health Department staff that actually go and collect the samples on a weekly basis. In terms of a timeline, we started the whole program in March of 2020, and schools were added in December 2020, as I mentioned, initially just for SARS-CoV-2. In September of 2021, we started monitoring for influenza in wastewater samples as well. And then in September of 2022, we added RSV. And then most recently, we've expanded targets to include measles, mumps, and rubella, and then also enterovirus D68. We actually started influenza and RSV at the facility level before we started monitoring at the citywide level. And due to the success of monitoring at this facility level, we expanded it to the citywide monitoring. So now we have, quite a bit of data, as I showed on the last slide, 3.5 years, 2 years of data for flu, influenza. And so here's some of the data. So we report the detections at the facility level as a presence or absence. And this is instead of a trend like we do for the citywide levels. And this is in part because there's a huge amount of variability, a lot more variability in the concentrations that we're detecting at the facility level as compared to the downstream wastewater treatment plant. And so this is a heat map. I know it's totally illegible, but basically the y-axis is all the schools and the x-axis is time. And this is showing our last school year. And this is for SARS-CoV-2. So if we had a detection in the school wastewater, it's shown as red. If it was below the detection limit, it's showed as green. And then if it's inconclusive, which is basically, usually it means it's right at the detection limit. We do replicate samples. So one sample's positive, one sample's negative, it's shown as orange. And so I'll show the rest of the data for the additional targets that we brought on. So influenza A is in the top right, B in the bottom right, and then RSV in the bottom left. As you can see, SARS-CoV-2, as others will also note, is much more abundant in wastewater as compared to these other respiratory viruses. But we did see detections of these other viruses that really corresponded to the waves of the respiratory viruses in the city. So you can kind of see the wave of RSV passing through the city and the schools, and then also for influenza A and B in the schools. And so we've done some analysis on this data, and we published a paper on it, which I'm not going to talk too much about, but it basically showed that if we looked at the SARS-CoV-2 data really early on, especially kind of in the early days where there was a lot more clinical testing, we saw that the detections at the school level were reflective of not only cases within the school, so we looked at case data where people actually were tested within the school, but they were also reflective of cases within the community. So there was a significant positive association between detections in the school and the zip code positivity rates of where those schools were located. And so this suggests that wastewater surveillance at schools can tell us not only about what's going around the school, but also kind of what the levels are in the larger community. And so we also looked at influenza data, most recently the 2022-2023 school year data. We looked at influenza detections across the school. And so this plot is just showing the proportion of schools that had positive influenza A detections plotted against the syndromic surveillance. So this is case data. So these are discharge diagnoses of influenza collected via syndromic surveillance. And again, we see this significant and positive association between the wastewater detections in the schools and what we're seeing in terms of citywide influenza rates. Okay, and then actually I think I changed my slides at the last minute, but I didn't get updated. But this is something I guess I'll talk about now. This is kind of some of our newer data that's showing how we've used this data to look at other potential inputs of influenza into the system. And I know one of the really hot topics right now for wastewater surveillance and interest is influenza A and H5N1. And so this is actually showing all of our longitudinal data for influenza A across the city. So time is on the x-axis, and then the pink line is showing the citywide wastewater viral load. So this is what's collected at the wastewater treatment plants. And this is the aggregate amount of influenza A across Houston from the wastewater treatment plants. And then the black line is showing the percent of schools that are positive over the same period for influenza A. So as you can see, we started our influenza A monitoring at the schools before we started citywide, which is why the black data goes back further in time. And you can see through the flu seasons, these different peaks. So you see, especially in the 2022-2023 school year, which is that middle peak. I don't know if this has a laser. Yeah, this one, here we go. They actually really correspond well to each other. We see a clear peak in the schools and also in the citywide wastewater viral load in terms of influenza A levels. Interestingly, we really did not see that peak in this most recent flu season. We saw the peak in the citywide wastewater viral load, but we did not see it in the schools. But we did see this second late peak of influenza A, which corresponds to when we had that kind of outbreak of H5N1 across Texas, and other groups such as TEFI in Texas, and also Wastewater Scan reported positive detections of H5N1 in Texas wastewaters. And so we see that in the citywide data, but we also see this kind of peak in the schools. And we actually think our hypothesis is that, as others have suggested, these are inputs from dairy. We see that school milk is served at every school lunch. Who has kids, that's all they get is milk as a lunch drink offering. A lot of that milk goes down the drain. We even have noticed in our wastewater samples we're getting in the lab a milky color to some of the samples. And so we think that some of these detections, because we see no corresponding syndromic surveillance for this peak, are due to these dairy inputs into the wastewater. And so this is just to suggest that school surveillance could potentially be used to understand inputs of some of these H5N1 or other influenza A inputs into the sewer system. Okay, that's a little bit of a tangent. I'm going to go now into talking about how these results are communicated to different stakeholders. So this is a list of the stakeholders that receive this information every week. And we've really focused in this last year on expanding and improving our communication from our school surveillance work. And on the bottom is kind of some of the visualizations associated with these reports. So this information goes to health department leadership and city leadership, school nurses and outbreak teams. And in this past year, we've really focused on getting this information also to student caregivers through a school virus alert program. And so now we've had this program in place for about 1 year now, and we're excited now that today's the first day of school to start this program again. And so the school virus alert program is just what it sounds like. It's an alert system where you can sign up for push notifications when there's a detection of any of these respiratory viruses in your school. So you can subscribe for one school or multiple schools, where if we have a detection in the wastewater, you get a push alert notification saying, look, we detected this in the school, and here's some information about where you can get tested and or vaccinated. And it's provided in both English and in Spanish. It's been a highly collaborative effort, really led by the health department. They work with the city Office of Emergency Management to design this alert system. They're the ones who actually send out the push notifications. And the success of this is really largely also due to the community involvement coordinator, who's gone school to school and gotten people to sign up for this. So we have links provided, but actually the majority of the signups for caregivers and parents has been because this person has gone kind of and individually signed up parents at each of these schools. And overall, the feedback has been super positive. School nurses have said they've used it to think about when to do sanitation efforts at the schools. Parents and caregivers have said it helps them have some idea of what's going around the school. And if they have an immunocompromised person at home, they can take extra precautions or make sure they get that vaccination that maybe they were putting off. I think most importantly, we've used it to do targeted vaccination events. So we've had 2 positive consecutive detections of influenza at any of these schools. The Houston Health Department offers a free vaccination clinic at that school where they say, here, you can get your flu shot, you can get your COVID shot. And while you're at it, you can also get updated on other routine vaccinations. So we've also seen some, caregivers are choosing to get their kids updated on other vaccines that they're just behind on because life, which has been great to hear. So numbers to date, we've had about over 5,000 alerts sent out based on these results. Over 1,000 individuals have signed up, and 7 vaccine events have been hosted. We've learned a lot from this first year, so we're really excited to continue it this year and in an improved version. We know that this in-person promotion has been really critical. This is just a picture of the little kind of booth that's set up outside these schools to get parents to sign up. Using accessible language is really important, and taking out some of the jargon that I know I use all the time that makes this like demystifies wastewater surveillance. And then I think this is the last slide I have, which is say we're really focused on expanding surveillance to other really actionable disease vaccine preventable targets. Measles has been, many outbreaks have been reported globally and within the US over the past year. And so we've been kind of bracing ourselves for quite a while for a potential measles outbreak within Houston. And so we started by developing a multiplex assay that detects measles, mumps, and rubella using RT-ddPCR. And then we also developed an assay that distinguishes between wild-type measles and vaccine strain measles, because this vaccine is administered as an attenuated live virus. So you can't, well, depending on how you design your assay, you can't necessarily distinguish between that vaccine strain and the wild type. And we've implemented this surveillance now, I think, since February. We are happy to report we've never detected any wild type measles in our schools or in our larger citywide surveillance. We have had very, very rare, I think maybe only 1 or 2 detections that were vaccine measles, and we confirmed those using sequencing. So we have detected our vaccine, but we developed these assays. We validated them not only on standards spiked in wastewater, but also we received some wastewater samples from a collaborator in Chicago from a facility where there was an active outbreak. So we were able to show that this assay is able to tell and distinguish between wild type and measles vaccine. We have a poster on this, so please come chat with me if you're interested in learning more about it, and also a preprint for some of the details. And so with that, I will just say, here's our contact information. We are a center of excellence, and so we love working with everyone in our jurisdiction and collaborating on assays, protocols, best practices, and exchanging samples for assay validation. So thank you very much.
Thank you, Lauren. I'm always so surprised to hear how many sampling events are happening at Houston. There was a lot happening in the beginning of the pandemic, and I'm like thrilled to hear that this is still continuing. So There is, I just want to, I'll mention this later as well, but there is a mic in the middle of the room here for questioning. But we will hold the question until the end. So our next presenter is Dr. Edwin Oh. Dr. Edwin Oh received his PhD in neuroscience from University of Michigan at Ann Arbor in 2008. Following a postdoctoral fellowship at Johns Hopkins University, he served as an assistant professor in the Department of Neurology at Duke University. Dr. Oh is currently tenured professor in Kerkorian, maybe I'm messing that up here, School of Medicine at UNLV. His current research program involves screening wastewater for the presence of the pathogens such as the SARS-CoV-2 and influenza and sequencing the RNA of these viruses to contribute to future vaccine development. And here is an interesting fact. He's also an expert in schizophrenia, autism, and addiction. Dr. Oh?
Thank you so much for that kind introduction. Hello, everyone. My name is Ed. I'm a professor at UNLV, and it's a real pleasure to present a few short stories to you today about mpox and Candida auris. So before I start, I always like to acknowledge the group that we work with in the lab. We have the privilege of working with very, very highly motivated undergraduates, and many of these individuals play a big role in sampling from all of the different locations across Las Vegas. So this is at dormitories, at elementary schools, at the Las Vegas Strip, at bars and hospitals. And if it weren't for the 20 or 30 undergraduates that we have, we wouldn't be able to sample upstream. I've also been very, very lucky to be mentored by a number of environmental engineers, because 4 years ago, I had no idea about anything to do with wastewater or manhole covers. And so it was a real privilege to understand how to open up one of these manhole covers and look at this amazing resource that we can now use to sample, to look at a number of different microbes and also chemical analytes. And really, it's Dr. Dan Garrity who's played the mentor for me in this role. I've also been privileged to work with the health district. I'm trained as a neuroscientist, so we didn't really know how to translate a lot of the data that we were getting to various actions for epidemiologists. So working together with the health district in Clark County in Southern Nevada, and also the state helped us be able to take this data and think of the various interventions, testing, vaccination, protocols and programs that we started developing. A lot of what we do is also the result of a public-private partnership. And I think it's really this interaction with a lot of the different companies that has helped us get far better with the technology in detecting these microbes and also in sequencing these microbes, because this is a major interest for us. And we're also very lucky to have the funding available to us. So most of what I'm going to tell you happens here in Nevada. We are a state of about 3 million individuals. About 600,000 people live up here in northern Nevada by the capital, up in Carson City and Reno in Washoe County. And a lot of us live down here in southern Nevada, about 2.3 million people. And a lot of our stories happen here in Facility 1. And part of the reason for that is because most people, when they arrive in Las Vegas, end up here in the Las Vegas Strip, right in the middle of paradise. And we have a number of upstream locations that we can sample from that helps us to isolate regions in the Las Vegas Strip, bars, and compare these data points to what we are seeing at the treatment plant that I'll be calling Facility 1. We can also look at other jurisdictions like Facility 3, which corresponds to the city of Las Vegas, city of North Las Vegas, Henderson, and Boulder City. And a primary interest for a lot of our projects is really threefold. We want to be able to use wastewater to identify the microbe of interest. We also want to use wastewater to be able to sequence these microbes of interest. And the third is we want to be able to develop interventions that can help us be able to address these different microbes. This is also true for chemical analytes, where we're now working with various universities to be able to look at different opioids. So the upstream sampling locations include hospitals, they include the Las Vegas Strip, tunnels under the city of Las Vegas, bars, and the airport. And we compare a lot of these data points to the corresponding wastewater treatment plant. So today, I'm going to be talking about a story about mpox at strategic sampling points, and also at hospitals where we've been looking at Candida auris. So this story is really the result of a talented research assistant professor in the lab, Dr. Van Vo, and a graduate student, Michael Moshe. So again, we have the privilege of working with a health district and looking at public health surveillance of a infectious disease of interest. And back in 2022, like in several cities across the country, we were seeing an increase in the number of cases around the end of July to the beginning of August. And so we wanted to ask whether wastewater surveillance could give us some intelligence about these different cases and what zip codes we should be targeting our interventions. And so we ran wastewater surveillance for these different locations across the facilities that I had just shown you. And we were detecting mpox viral DNA in these different facilities as early as September of 22. And these results were extremely frustrating to us because, as I've shown you earlier here, many of the cases were being detected towards the end of July and towards the beginning of August. So it wasn't until we started sampling from these upstream locations that we're calling strategic sampling locations near the strip that we started to see some corresponding data of the wastewater to the clinical cases. So we started focusing on a lot of these different zip codes. So we were able to detect mpox. We then wanted to be able to sequence this pathogen. And as you know, back in 2022, there wasn't a amplicon sequencing panel that could help us sequence this pathogen. Now, SARS-CoV-2 is about 30 kb. mpox is 200 kb. So we needed to design a panel that could cover something that was 6 times bigger in terms of genome size. And so working together with the health district, we were able to look at these various genomes, the distribution of genomes in 2022. Now, the number of samples here aren't too many. We sequence about 30 or so clinical samples. And here's the distribution of those clinical samples. When we compare that to the wastewater genomes that we were identifying, we didn't really see too much of the intelligence or early warning of the wastewater genome sequencing at the subclade level, where folks are using this nomenclature of B.1, B.1.2. However, when we started looking more specifically at variants or mutations in the genome, we started to note a number of interesting findings. For example, in this case here, we were able to identify a wastewater sample on August 14th for a mutation, serine 149 leucine in the ankyrin-like protein 3. And this is a very, very rare mutation. In August, we did not find this mutation in various databases. Now, when we looked at the clinical cases in Nevada, we identified a clinical case in September 7th. So this was somewhat early warning in terms of identifying the mutation earlier in the wastewater sample than in the clinical sample. And another interesting feature here is that when we started looking at NCBI virus collection, we also found this mutation later on in Texas in September of 2022. Because this is such a rare mutation, we think that this might actually be 1 or 2 individuals that were potentially present in Las Vegas in early August, and then ultimately were tested in Nevada in September and or in Texas also in September. We could also look using this type of data at the variation that might be present in genes of interest that are relevant for antivirals. So an antiviral for mpox is TPOX. And when we start seeing mutations in this gene F13L, then we'll want to be concerned because this antiviral treatment may not be as efficacious. So the equivalent here is that we have many of these vaccines against SARS-CoV-2. If we start to see mutations along the spike protein, then potentially the vaccine may not be as efficacious. So this is part of the reason why we start looking at the F13 gene. And we could identify this mutation here, this glutamic acid to lysine mutation here, in all of the clinical samples and also all of the wastewater samples. So this gave us more confidence that we were finding the right thing. And we start seeing these novel mutations in F13L that has now given us the opportunity to look at modeling to see how this might affect the role of TPOXX and whether it's efficacious. So we're able to identify mpox, we're able to sequence. How about interventions? So in terms of vaccination sites at the health district, there were about 45 different static sites across the county, Clark County. And these individuals don't move. They're in their offices, they're in their clinics, and they administer these vaccines. We compared these traditional vaccination efforts with mobile strike teams that were deployed as a result of this wastewater data. And we were quite surprised to see how efficient these strike teams were. We were able to deploy 8 different strike teams, and with 20% of all of the resources that were typically present at these traditional sites, we were able to administer just about the same number of vaccines across Las Vegas. I didn't think this would work in the beginning because these strike teams were going out at 10 p.m. and they were out till something like 6 a.m. So what's the possibility of going out in Las Vegas, going to a party, and then thinking to yourself, maybe I should get a vaccine? Apparently, it works. So here's the data. So given that we were able to identify this pathogen, we wanted to use these different strategic sites, sampling sites, to look at other pathogens. And we gained more confidence from the various sequencing efforts to look at other pathogens. And so I'm going to quickly go over some of the data that we've seen for Candida auris, because I know there are a few other talks during the symposium addressing this pathogen. And this is a project that's led by a talented graduate student, Lanny Chang. So for whatever reason, in Nevada, we have seen a very high number of Candida auris infections. And when you look at all of the different cases across the last few years, about 60% of the 3,000 cases or so have been found in males. And about 80% of all of these cases are found in individuals who are older than 50 years old. If you now look at all of the different clinical cases and you plot this here in B from 21 to 23, what you can see here is a bar that corresponds to every month. And what you're seeing here in black is the presence of this pathogen potentially in the bloodstream. What you're seeing here in this light gray is colonization by this pathogen. So this pathogen is present on skin, and so we record this detection. And so what we ended up doing was sampling around this time point from July of 23 all the way to the end of the year. And we were primarily sampling from 3 different hospitals. So here's the distribution of cases of clinical and colonized cases down here in C. And really the first result that we noted that was quite interesting was at the level of the wastewater treatment plant, we didn't really detect Candida auris at a high frequency, either from this 4A location or this facility 1 location. But when we started looking at the wastewater from the various hospitals, the frequency of detections was very much higher. So we wondered to ourselves, how could we use this information for interventions? One of the concern in the field for detecting Candida auris in sewers is that this is a pathogen that might be growing in a sewer line. So is it possible that the frequency, the higher frequency of detections here is because this is a pathogen that's growing in the sewer line, and therefore we're just detecting a pathogen versus the number of cases from the hospital. To address this question, here are some data which you probably cannot see, but what I'm hoping to show here through amplicon sequencing from the group is that we designed a target that was about 30,000 nucleotides to look at key genes that were present in Candida and mutations that are usually key in Candida and oftentimes Candida auris. So here's a mutation valine to alanine 125. And so we were looking over time, September 7, 12, 14, 19, at the frequency of detecting this mutation. And what this is showing you here is that 4% of all the sequencing reads corresponded to this alanine. September 12, there was no alanine. And then 14 was 33% of those reads were alanine, 12%, and so forth. So we knew that we could track some of these mutations, and they gave us a little bit more confidence that it wasn't the same genome that was present in the sewer line. As I mentioned to you, we looked at 30,000 nucleotides, and so we could go across one hospital and look at these different mutations that we found to be very interesting in Candida auris. And again, the frequency, the allele frequency of these mutations was very much different across time. And not only were they different across time, they were also different across space. So in different hospitals, we were seeing the frequency of these mutations change over time at different rates. So this gave us a little bit more confidence. But we wanted to go a little deeper with whole genome sequencing. We wanted to take that wastewater sample, bring it back into the lab, culture Candida auris, and then pick individual isolates and sequence the genome. SARS-CoV-2 is 30,000, mpox is 200,000, Candida auris is about 5 million. So we looked at the whole genome, and here is a phylogenetic tree. And the information that I really hope that you'll gain from this is when you look at clinical samples that are in pink and blue samples that are in wastewater, there is very much a relationship of all of these different samples. We compared our wastewater data with the 3,000 genomes from clinical samples from these various hospitals. And when you look specifically at a branch, here is a subclade, which we call clade 1.A.1.C. And the reason I highlight this and another subclade is because if you look at this wastewater sample here, it was collected October 10, 2023, October 10, 2023. These clinical genomes weren't identified until November or February, November of 23 or January or February of 2024. So wastewater was giving us the opportunity to identify these genomes before the identification in clinical samples. The same was true for this other subclade, where we're identifying these genomes around September of 23, and the clinical genomes were showing up in the different hospitals later in time. So that gave us confidence that wastewater could give us early warning information about these clinical genomes. And here's a really, really interesting thing. Again, this was something I also didn't believe was going to happen. 90% of all our samples were within 3 shared SNP from the closest clinical isolates. So what we're seeing from the wastewater is very, very similar to what we're seeing in the clinic. So finally, given that we can culture these different isolates from wastewater, we wanted to understand antifungal resistance. So here's a sample where we can culture Candida auris, and they're susceptible to the kinocandins. What we're finding from wastewater now are these types of genomes where the samples grow even when you start throwing on high doses of mycofungin. And the interesting thing here, and the unfortunate thing here, is that these genomes here are now showing up in the clinic, where we're starting to see individuals with these genomes that are completely resistant to all antifungal treatment. And if you're resistant to all antifungal treatment, there's a high chance that there's a high chance that you'll be staying in the hospital for a little longer. So this gives us an opportunity to go from a service, the wastewater surveillance, back into the lab to identify drugs that can kill this Candida auris. And I'm going to skip this part. This was updated last night because I think I'm over time. So what I'm hoping to show you really is that you can take a lot of these different isolates, you can grow them at 42 degrees Celsius, compare them to 37 degrees, and look at pathways, look at genes that might be relevant for the growth of these different genomes. So finally, what I think is really, really important from a lot of the case studies that we've been looking at is that in order to do what we've been doing, collaboration was key. Understanding how those sewer lines were connecting to the treatment plants. If it weren't for these maps, we wouldn't be able to understand where we could sample from. For example, most recently, Carmel Harris was in Las Vegas. So where we were sampling from at bars, we had to move our auto sampler to different locations because the whole area was locked down. So working together with the environmental engineers, we were able to get this intelligence and be able to respond. And the other two things that I think are really quite key is that upstream sampling has increased our odds of being able to identify these pathogens, culture them, and also sequence them. And thank you. And I think we're going to move on to the next talk, right?
Thank you, Dr. Oh. Slightly frightening information on Candida auris, but I think this is an opportunity for us to all think about what's coming up and the utilization of wastewater surveillance. I think the last two presentations really showcased how we can utilize wastewater surveillance and really focus on public health action. So our next presenter is Dr. Cindy Friedman. Dr. Friedman is the founding director of CDC's Traveler-Based Genomic Surveillance Program that utilizes voluntary traveler nasal testing, aircraft wastewater, and air sampling at several major U.S. international airports for early detection of priority pathogens and SARS-CoV-2 variants. She serves as a technical advisor to GLOBOCON, the EU Health Emergency Research Agency's global consortium for wastewater and environmental surveillance for public health. Dr. Friedman came to CDC as an epidemic intelligence service officer after training as an infectious disease physician at Cornell Medical College. She spent several years leading CDC's National Antimicrobial Resistant Monitoring System and holds an adjunct faculty appointment at Emory University's Rollins School of Public Health. Dr. Friedman.
Okay, good morning, everybody. As Anna mentioned earlier, many developments in biosecurity and biosurveillance have emerged from the COVID-19 pandemic. And so today I'll discuss one of these, which is airplane wastewater surveillance. This film illustrates a key lesson from the pandemic that I think we can all remember, that an outbreak anywhere is an outbreak everywhere. And the movie depicts a single epidemic showing carriers that are traveling through a single flight connections, and it's courtesy of Alex Vespignani and his team at Northeastern University. So knowing that travelers are an important population to consider when we're tracking new and emerging infectious diseases because they move place to place, we have known for many years that we use clinic-based sentinel traveler surveillance for public health, and we've been successful for decades. But that approach requires the traveler to be symptomatic, seek medical care, go to a clinic. The clinician needs to order the right lab tests. Those lab tests need to be reported, and then genomic sequencing needs to be done. So this takes a lot of time. And when COVID emerged, it was missed by the existing traveler-based clinics, the clinical surveillance systems. And because air travelers move so quickly that when a new variant of SARS-CoV-2 emerged in one place, it had already spread globally. So during the pandemic, CDC leveraged a public-private partnership, and I know we've heard that in some of the other talks, but this one was with an airport spa company, ExpressCheck, and a biotech company, Ginkgo Biosecurity. And we developed a traveler-based genomic surveillance system for early detection. And if I had more time, I'd tell you the funny story of how this all happened. But if you want, you can read it in the Bloomberg article. And using this public-private partnership, we were literally able to stand up our monitoring system over a weekend during Thanksgiving in 2021 when Omicron hit. And I don't think that would be possible without that partnership. So I think we may disagree about what decisions work best during the pandemic, but I think one strategy that we can all agree on is the value of early detection. And speed really helps buy that critical time that we need to make public health decisions. And it can really also help avoid costly border closures and travel and trade restrictions that we saw during the pandemic. So since its initiation in September in 2021 as just a small proof of concept project, the Traveler-Based Genomic Surveillance Program, or TGS, has grown and evolved. It uses a multimodal platform to achieve two main goals, to enhance early detection and to fill gaps in global disease surveillance. The platform is sample agnostic, so we can use it to look at multiple pathogens and can use the best modalities for the specific pathogen or target. We have different modalities listed there, and just briefly, TGS collects nasal samples from arriving US international travelers at airports. No personal information is collected, and it's completely voluntary and anonymous. We just ask a few questions about travel destinations and demographics. The whole process takes under 5 minutes, and we can collect as many as 9,000 samples from volunteers per week. But this approach is really labor intensive. You can imagine that it's costly, and it involves a large staff presence at the airport. So in early 2023, after a successful pilot project at JFK Airport in New York, we added aircraft wastewater sampling to the platform. Airplane wastewater provides early detection without direct traveler involvement or disruption to airport operations. We can sample about 300 to 500 people with just one airplane wastewater sample. And it is an early proxy for what is happening in the community. Results, including genomic sequencing data, are usually available within 10 days of collection. Sequences are shared on global databases, and the raw read sequences are uploaded. Am I getting feedback? I hear like oh, where did it go? Sorry. Maybe this wasn't bad. Yeah, okay. I'll just hold it. I'm good. No, it's good.
Just put it on your clip.
Where did it go? Sorry. I'm trying to strangle. Okay. Okay, so sequences are available in 10 days, and they're shared on the global database from the National Center for Biotechnology Information, or NCBI. This is the map of the airports where testing occurs. And for the nasal program to date, we've tested over 600,000 travelers since inception from over 130 countries. So we've achieved broad geographic coverage. And since February of 2023, when the wastewater began, we've collected over 1,000 airplane wastewater samples. Since December of 2023, we've began testing for multiple pathogens, including SARS-CoV-2, flu A, flu B, RSV, and mpox. So today I'm just going to focus on the two points of collection, the aircraft wastewater and the triturator wastewater. Triturator is a consolidation point that captures wastewater samples from multiple flights, but doesn't include the airport terminal waste. So I just want to review the flow of waste from the plane lavatory tanks. So when the airplane is on the tarmac, the ground handler removes the waste from 3 to 4 tanks that are on the plane. It depends on the type of plane. Waste is extracted through a hose, and I'll show you some pictures in a minute. And then it's combined with waste from several other flights in this laboratory truck. And when the truck is full, after around 4 to 5 flights, it's emptied into the triturator or that drain collection point. So to collect wastewater directly from a plane, we use a custom collection device from Ginkgo Biosecurity, one of our partners, that attaches to the hose used to extract the waste from the plane. The device has a side spigot that you can see in the center picture, where a 1 liter bottle is attached. The valve on the spigot is opened and some wastewater runs into the bottle. The ground handler takes a sample from each tank on the plane, and the device is cleaned between uses and is transported in this case that you see on the bottom left, on the yellow case, out of the airport. Samples are shipped to the lab. And the whole process really only adds 2 to 3 minutes to the normal operation of the ground handler. And we have a staff member on the tarmac that gets the bottle and takes the case. And so the other collection point that I mentioned was the triturator drain. So when the laboratory truck is full, it dumps it at this drain. And we use an auto sampler that attaches to the triturator and does not disrupt any normal operations. Its flow activated, so it can be left unattended for days without servicing, and samples are retrieved 5 days per week. So let's look at some data. So starting in December 2023, we began the multi-pathogen testing. And here I'd like to point out some of our results for some key respiratory pathogens, including RSV in the purple, flu A in the blue, and COVID-19 in the orange. And for comparison, in the small box on the side is the respiratory activity levels based on weekly clinical positive tests for the same time, approximately the same time period. We can see in the wastewater graph, the big graph, that flu and RSV positivity is high in the winter, and by spring, it declines, as we would expect from these seasonal viruses. SARS-CoV-2, as we know, has a different pattern that has peaks both in the winter and the summer. But we did see a decline in this spring from around late March through May. And then you can see it goes back up for this surge that we're all living through this summer. So the airplane wastewater and clinical testing curves, though on different scales, look similar and show similar trends. The genomic sequencing raw data for SARS-CoV-2 from wastewater are, as I mentioned, reported to NCBI within around 10 days of collection. The team looks at our wastewater samples for new variants of SARS-CoV-2 and looks for mutations with increasing frequency independent of lineages, allowing for detection of novel emergent mutations early. The graph on the left shows SARS-CoV-2 lineages detected in our wastewater from February 2023 to July 2024. And we've detected over 17 major and minor lineages. We also track concerning mutations like the FLIRT mutations shown in the graph on the right, the graph on the right in green. And FLIRT and FLIP mutations were used to describe the family of variants, mainly KPs and JNs, that have independently picked up the same set of mutations. This mutational profile represents most sequences reported publicly over the last few months and is really associated with this summer surge that we're seeing. So FLIRT mutations were picked up in our airplane wastewater samples in mid-November, really quite early, before any global reporting, and about 1 month before a sample was collected with those mutations in the US, and that sample wasn't reported for 3 months. So this really highlights the early detection, the timeliness, and the value of our program reporting to those public repositories. And this is an example that I'd like to show that really shows the impact of wastewater surveillance and the complementary approach. So this is an example from Boston of how airplane wastewater can be the leading indicator for surges in the community. And this was work done in collaboration with Secure Bio in Boston and Ginkgo Biosecurity. So from November 3rd to December 22nd in 2023, 140 wastewater samples were collected from Boston Logan Airport Triturator and the Boston wastewater treatment plant at Deer Isle, which captures the community. The data were compared with public database reporting. And on November 7th, BA 2.86 was detected in a single sample of airplane wastewater from the Boston Logan Triturator. And as a reminder, BA 2.86 first appeared in August 2023. We wrote an MMWR about it. It was a new SARS-CoV-2 Omicron subvariant with many mutations compared to previous circulating variants. It had like 30 mutations. And it emerged in multiple countries almost simultaneously, but it didn't really take off right away. We kept waiting. We even had bets, like, is it going to be the next new variant this week, next week? But it took a while. And even in October and November, the previous variant, EG5, if anyone remembers that, was still dominant and being reported to GISAID, the global repository. But on November 20th, WHO declared BA 2.86 a variant of interest. And if you look at the curve on the right, in Boston, by December 6th, we are seeing in airplane wastewater, 2 weeks before the surge appeared in the community, a surge in the BA 2.86 variant. And that was before the wastewater and community cases surged in Boston. So this really, what is this telling us? That aircraft wastewater can provide an early signal, and it can be used to detect increasing trends of new variants before an uptick is detected, even in community wastewater and clinical monitoring, and especially when variants emerge maybe from outside the country and come in. And this really also shows that there's a real role for airplane wastewater, community wastewater, and clinical monitoring. So they're all complementary. And if we use them together, we can have a more complete picture. And it sort of supports the idea for super sites or centers of excellence for wastewater surveillance, where the airport, the school, the hospital are all the sampling's integrated. So there are some limitations associated with airplane wastewater sampling. Not all travelers use the laboratory on planes. We know that from some work done by Davy Jones at Bangor University, that the chance of defecation is around 36% on long-haul flights over 6 hours. Airplane wastewater may not really reflect the true country of origin of variants or pathogens detected, because not all travelers originate where the flight originates. They may have had 3 connections before that. But I think with multiple geographic sites collecting airplane wastewater and using the existing flight data, we can determine the country of origin of variants and pathogens detected. It just takes a little math and a little work. Airplane wastewater sampling requires close coordination, and this is probably the most challenging piece of this puzzle. The airline agreements, the airport agreements sometimes can be time-consuming and daunting. We've spent a lot of time working with them, explaining the value, but it is something that needs to become more routine. Not every pathogen can be detected in wastewater. That can be detected in wastewater is a good use case for public health. I think I'm preaching to the choir here. We all know a lot about this subject. There are decision support tools that are being developed by WHO and CDC that can really help with this. Just because you can find something in wastewater, it may not be a good use to do it in airplane wastewater. And definitely more work is needed to determine limits of detection, positive and negative predictive values for certain pathogens. You know, one of my mentors used to say, and I think it's a quote from Carl Sagan, absence of evidence is not evidence of absence. So sometimes if we're not finding anything, I'm not always sure that it's not there. So for the last few minutes of my talk, I'd like to just switch gears and talk about some global efforts that are going on with wastewater surveillance in airplanes. So let me just start with some elegant modeling work from Alex Vespignani's lab at Northeastern that demonstrates the potential of a global airplane wastewater network for early detection and pandemic preparedness. They used a GLEAM model, and I'm not a modeler, and it integrates mobility data with spatial and temporal data to describe the evolution of an emerging epidemic. And they use this model to provide estimates for the spread and detection of a pathogen using aircraft wastewater surveillance. This GLEAM model has been validated for various health emergencies, including H1N1 and Ebola. So this is a retrospective look at the alpha variant B.1.1.7, which was first detected in the US in January 1st, 2021, after emerging in the UK back in September before that in 2020. And it was sort of missed here. It took a while for us to find it. To evaluate the performance of a wastewater surveillance network like the US-based Traveler program, their model focused on time elapsed since the beginning of the alpha outbreak to obtain a first detection. And I can't go into all the details, but the bottom line is they showed that detection used with our program in place at that time would have had a detection 1 to 2 months before the actual detection occurred. And then if they further looked at what if we had a global network with 10 to 20 well-placed wastewater sentinel sites, could that serve as an effective early warning system to improve detection, even if there were some geographic gaps in this system? And they showed through that same retrospective look at alpha variant that they would be even a bigger, there would be even an earlier detection, 2 to 3 months earlier. So several other models have shown similar findings, and there's much global interest in putting this model to test. And since I'm an epidemiologist and like data, we're actually going to do this in September, which is really exciting. And the European Commission's Health Emergency Response Agency launched GLOACON, which is a global initiative to create and maintain a global network for the implementation of wastewater and environmental surveillance for public health. So this coming September, we're launching an airplane wastewater pilot that will include synchronized collection and coordinated examination of wastewater samples originating from aircraft at a variety of global airports within a controlled framework. And for this first pilot, the overarching objective is to foster the establishment of uniform methodologies and standard operating procedures. And to add on to that, in addition to the multilateral efforts, CDC is working with the UK HSA, the UK Health Agency, for a 6-month proof of concept pilot to further our knowledge and capability in wastewater monitoring for planes that are traveling between the US and the UK. We're excited to expand these efforts and make global collaboration for biosecurity and preparedness more routine. And this is my last slide. So imagine if we had traveler-based genomic surveillance in place in early 2020. Early and rapid detection can buy critical time to institute appropriate public health measures and avoid costly border closures and travel and trade restrictions. So I'd like to close with the 5 M's that I believe are key to the success of a traveler-based program. And the first is that traveler-based genomic surveillance leverages multimodalities, and this approach can be flexible and adaptable to a changing landscape. The one platform approach can be used for detection of multiple pathogens, which saves resources. A global airplane wastewater network in 10 to 20 global locations can substantially decrease the time to detect an outbreak or a new variant. And this is critical time save, but it's going to require global cooperation and data sharing. And everyone needs to have a chip in the game. A whole society approach with multi-sectoral partnerships, including airports, airlines, ground handlers, is essential. And perhaps the most important is that during these quiet interpandemic or somewhat quiet interpandemic periods, the challenge is really to maintain the platform and keep the system warm so that it can be scaled up quickly when it's needed. And I hope this really shifts us from a cycle of complacency followed by crisis to one that is committed in maintaining the best tools for biosecurity. Thank you. And I just want to acknowledge, I couldn't list everyone's name, but there are so many people that really make this whole system happen. So thank you.
All right. Thank you, Cindy. And it really resonates to me what you just mentioned. We really have to have a good understanding of a clear sampling design plan with sentinel sites. And I think that's the really important next step. So okay. So Scott Bessler, he serves as an interim superintendent and chief of the Compliance Services Division of Metropolitan Sewer District of Greater Cincinnati. He holds degrees in biological sciences from Northern Kentucky University and University of Alabama. His MS. He also oversees strategic research and initiatives for the district with a focus around emerging contaminant and pathogens and beneficial reuse of waste streams and general water quality. Scott?
Hello, everyone. Can you hear me? How's the audio in the room?
All good.
Very good. All right. I've been notified that I have a time restriction, I believe, so I will try to fly through this. That's my joke about the last talk. Ha ha. All right. So hello, everyone. So excited to be here to talk to you about my new favorite term, which Dr. O threw out, wastewater intelligence. And you may ask yourself what that means. Well, to tell you the truth, I think that definition is evolving as we speak, but I'm here to talk to you about two activities that might fall under what I consider that umbrella. The first being infrastructure protection, which I'll talk about today, and the second being public health insights, which I believe everyone else at this at this meeting will talk about also. So I'll go ahead and get right into it. To begin, I want to stress the importance of partnerships between clean water agencies and public health authorities, and I think that this GIF and subsequent meme from the 1987 classic Predator sums it up well. This handshake between Carl Weathers and Arnold Schwarzenegger perfectly encapsulates the relationships that clean water agencies and public health authorities should develop. So this is representation that I like to use for our partnership, but really it's so much more. This is a partnership, I think, that is one that is needed in order to maintain and grow this exciting young field. And I'm really excited to talk to you about some work that we did around that here in the Cincinnati area. So the Cincinnati Health Department and the sewer district here in town completed a subsewershed study in early 2024 that measured disease burden in communities. But I'd like to talk a little bit about the dual benefits of carrying out this type of subsewershed monitoring. Not only do you gain public health insight into what's happening disease-wise at a neighborhood level, but you can also begin to understand what's going on within your sewers from a water quality perspective. And I do really believe that this is where the rubber meets the road for what I call the dual benefits of clean water utilities and public health authorities. So as a part of this study, not only did we sample for the public health targets of concern, but we also were sampling 4 different water quality parameters in real time. And it just so happened that in this area in red, I'll tell you a story about something that we found there that really kind of shows that dual benefit. All right, just to orient you about how water flows through this sewer basin that I'm showing right here. You can see my screen my cursor. Water flows from this side over here to the right to the left, down here to our Mill Creek interceptor, which is right around this area right over here. So general water flow from right to left here. So now that I've zoomed in on the neighborhood that we were surveilling for disease by the little red circle earlier, we're also monitoring water quality. And we discovered a number of really kind of strong, what we term pollution events that were happening outside the bounds of our sewer use ordinance. And one very specific parameter that we saw that was pretty wild swing was pH. So you can see down here is where we had our sampler in the bottom of the corner there. It's kind of hard to see, I think, but there's a auto sampler device and a sonde that's connected to that. And then I'll pull up a time series that we took from December. It's essentially like a 3-month period from December 2023 to January 2024. And what you have is pH that's plotted here. And you can see over time, you had fairly normal behavior of wastewater quality, but you saw these giant dips here of pH that were down less than 2 at times, which our sewer use ordinance only allows us to operate or have folks that are discharging into our system that have a pH between 5 and 12. So we had somebody that was doing something that they shouldn't have been doing. So if you know anything about how acidic conditions can cause issues in sewers, there can be a lot and if you don't know about it, I'll tell you about it, but there can be a lot of issues with acidic conditions and corrosivity in sewers. So after monitoring for nearly 6 months, we noticed these pollution events were happening at pretty regular intervals. And then we began to mobilize many sensors as we could out in our collection system to start to try to drill down where the problem was. So We moved up our trunk sewer here, and these red lines are the trunk sewers that I'm going up. I don't have a better illustration of it, but we actually got to the point where we traced pretty far up into the collection system, and we got into the upper reaches of the sewer shed, and we actually isolated the segment where the pollution that we had talked about much further downstream in the sewer had come from, and we confirmed it by placing sensors just downstream and having our TV crew record video on segments of pipe that we had isolated. So here's a couple of screenshots from that TV that we did earlier, and TV work by saying we put a camera down in the sewer. And what did that TV work show? Well, boy, there was some strong blue, purple wastewater shooting out of this lateral right here that you can see up in the corner here. And it was coming out. We didn't actually get it on video during this event, but you can see over here in the right hand or in the left hand pictures that there's pitting on the other side of this sewer across from where that lateral comes out. And so that the strength of that wastewater had caused issues from a structural perspective to this sewer. And even luckier, we caught, we had a camera installed down in the area where we originally had our first water quality sensor installed in our sewer. And, you know, I'll play a video here for you, and you can see that's normal colored wastewater if you didn't know what normal colored wastewater was. But then you can see this very stark color change that happens during that time period. And that corresponds with this time series down here below, where you see normal quality wastewater alongside here, and then a huge dip in pH, an increase in electrical conductivity, and an increase of oxidative reductive potential. And we were actually able to show that signature of water quality that we saw was visible not just from the eye from this camera, but also from the sensor that we had in our sewer. So it turns out that there was supposed to be this slug of water was supposed to be going through a pretreatment system, but the sewer on the property had leaked into another sewer and came to this point instead of getting treated. So they've since corrected this issue. So that's actually like a big win from our perspective. All right, so going back into time, this is the only other TV footage that we had. It was from 2009, and you can see that's that same lateral that was colored blue in the TV video that we had from 2004. But that's the other side of the sewer here. This is the corresponding picture of the 2004 TV, and you can see, oh darn, quick pass, there we go. The sewer is in pretty decent condition in 2009, and in 2024, there's considerable wear that happened over that time period. So how does this type of work, drilling up into the collection system like I just showed, benefit sewer utilities in terms of infrastructure protection? Well, should that discharge have been allowed to continue untreated, it would have possibly eaten a hole in the side of that sewer over here, and causing structural issues that could have resulted in the following. What we see here is a sewer cave-in that happened near the Cincinnati Zoo. And I'll go ahead and say that now no lions, tigers, or bears were lost during this incident, but you can see, and this made the news, that's where I got these pictures from, that we almost lost a city bus due to a sewer cave-in. So I wanted to kind of show you guys what a catastrophic failure looks like, and then talk about, from a cost savings perspectives, identifying these previously unknown industrial discharges will likely result in a point repair cost of roughly $50,000 to $100,000 instead of what could have resulted from this as a catastrophic failure, which was hard for our folks to estimate what the cost would that be, but anywhere from $500,000 to $2 million, depending on how catastrophic that failure was. All right, so. And I wanted to kind of just give some idea what the bigger impacts of these surveillance efforts are. This is that same camera. You can see there normal colored wastewater, and then it turns kind of green there for a second. You can see it right there as it turns green. I want to show you some of the bigger impacts. So this ultimately flows into one of our large interceptors that goes down to a 110 MGD plant. And then that green dye that you saw passing through there had this effect on our wastewater treatment plant. So that's 110 MGD wastewater treatment plant. And that dye that we saw that video of caused our plant to go from you would consider normal looking wastewater to this almost St. Patrick's Day Chicago River dyed kind of look to it. And then this is what our effluent looked like on that day. It was very green. And serendipitously, we had been going out of an outfall that does not go out to the Ohio River. We sit on our plant sits pretty close to the Ohio River, but we have an outfall that travels underground out to there. And you can kind of see it here, but we were going into an outfall that was going into a creek next to our facility, and it dyed that creek green. So that was just to give you kind of an idea of the larger impacts of what can happen, and understanding from a surveillance perspective or an intelligence perspective, what monitoring in their subsewer shed can do. So what do clean water agencies get? I kind of went over that. What do the public health entities get? They get the insights into public health in the subsewer sheds that you had isolated. So really there are dual benefits to doing this type of work. I kind of flew through that because I knew we were kind of running out of time, but I'll go ahead and I think we're going up into questions now. So I'll go ahead and stop. Thanks, everyone.
All right, thank you, Scott. That was an excellent case study of how subsewer sampling can impact both water quality information and public health. So right now we're ready for our Q&A. If all the presenters can come up in the front, that would be great, and we can start our Q&A. Again, a reminder, there is a microphone in the middle of the room if you have any questions, and we have someone else monitoring our chat. So if there are any questions virtually, we'll also be asking those. Okay, so I'll open the floor at this time for any questions. Go ahead. Please state your name, introduce yourself.
Hi, Scott Olson, CDC Center for Forecasting and Outbreak Analytics. There was one paper from some German folks about urine and showers and stuff. So Cindy, I was kind of thinking for you this thing about who defecates on a plane. I don't know if there's any other literature about this, but it sounds like a bad movie, right? But like, I think many people pee on a plane. I'm just thinking like what fraction of virus comes out in different places. And I don't know if that is something that other folks around this room too, it's not just for you, but folks around here, if we know more about that, I wonder if that will help with some of these questions.
So it's a great point. And, that study by Davy Jones looked at defecation on different flights. But we know obviously more people urinate on a flight than defecate. It probably depends on if the flight's an overnight flight versus a daytime flight. I think there's more work to look at that. And if anyone here has a great idea, I think that study was done a while ago, and we've been thinking about ways without invading privacy on how we could actually get at what people are using the bathroom, how many, and get more specific and repeat that. One thing, people also blow their nose and throw those tissues down the lav. So you don't only get urine and feces, so you do get other body fluids and probably some other things too. So we just don't have enough data as far as I'm aware, but I think it's worth looking at for sure.
Yeah, thanks.
Great. So my name is Prabhu. I'm from LA County. And this is a question for Dr. Stadler, because we're a large county. We're in the early stages of setting up our wastewater surveillance program. And one of the questions that I've been thinking about is the value of upstream testing, because it's really resource intensive, thinking about the number of sites. And looking at influenza as an example, typically the flu seasons are 12 to 16 weeks long. Once they start, it's pretty widespread in the community within 1 to 2 weeks. So I'm trying to think in that setting, when you do upstream testing in all these different locations like schools, how often do you see something that's different in an upstream location than what you would at the main plant?
That's a great question. I think that was one of our original goals of when we were analyzing our data, is to compare the upstream detections to the community detections. And so we definitely did see very similar trends and even timing. So when we looked at, this is one question we got, our school detections showing up earlier than community detections. When we looked at our 2022, 2023 influenza data, and I think also our SARS-CoV-2 data, we saw they were very concurrent with one another. We were not seeing earlier detections in the schools in the community, and we were seeing very similar trends. So, but then when I showed kind of the most recent data from the most recent flu season, and our most optimistic interpretation of this is based not in any real analysis of data, is that, oh, our school virus alert program is working really well, because all of a sudden we did not see the detections in the schools that we saw in the community. And so that was really this most recent year was the first year we saw the difference for influenza between those two, and we're not really sure why. So this is a great question. I think it also kind of goes to the actionability question. I think one of the reasons we really like the school and we really have invested in the school manhole program is because there's a clear action that comes from the detections, and that it can be used to target these vaccination events. Whereas I feel that with the citywide surveillance, it's almost a little bit more like it's a great source of information of what's happening trend-wise in the whole community, but it's almost like a weather report a little bit, and that people are looking at it to see the levels, less about like, oh, this is going around in my community, and therefore I'm going to go get a vaccination. And so I think there's a trade-off between the scale and the actionability of that information.
Thank you.
And just to kind of add to that, like having two small kids and they're always sick, and the exposure to grandparents, like let's just take RSV as an example. You want to limit that as much as possible and maybe warn your grandparents, hey, maybe you should go get RSV vaccine, So I think that's key for that kind of information for a parent.
I would say that's one of the biggest pieces, I think, of feedback we've had is people look at the wastewater levels to make decisions about seeing family, and even traveling to see family. Oh yeah, and maybe that's one of the reasons why people have been more interested in doing vaccinations when they know what's going around in their community, but they've really used that information to make decisions about who they see.
Thank you. Go ahead.
I've got a question for Dr. O as well as Dr. Friedman. Frank Pastor with Oak Ridge Associated Universities. So Dr. O, two questions. One's kind of a comment question, but on the strike teams and the vaccine uptake, any thoughts on liquid courage being a contributing factor to folks having that high percentage success?
A little unclear.
Okay.
Like I said, right? When I've gone out back in my good old days when I was young, I wasn't thinking of getting vaccinated.
Right.
and so there was a lot of hesitation, I think, and I think it was through the community partnerships that we realized that some of these types of actions can play a role in the interventions that we wanted to get out of it. And I think this was really where working together with the community partners and the confidence from the community partners that this was going to work. For example, a similar type of scenario, we tested for SARS-CoV-2 in tunnels under Las Vegas, where we know that there's a high percentage of unsheltered individuals. And so we ask ourselves, so why are we doing this other than potentially discovering what we already know? And the answer here is that we can now work together with other community partners to target some of these teams to different locations where we think there's a higher signal of the pathogen of interest. I think this is, again, where the community partnerships came in.
Thank you. And one other question for you on the hospitals and the Candida auris for Dr. O. Did you run into any challenges with these hospitals' willingness to participate when you're trying to set up composite samplers and say, we're just testing for this and there's no issues, nothing else is going to pop out of this? How collaborative were they willing to be?
Tremendous challenge, and for obvious reasons, right? Yeah. There's a great deal of liability here. And we were setting up this type of system to determine whether we could come up with actionable information, because some of these hospitals were thinking that we have the highest number of cases. It's normal for us to have Candida auris at our facilities. So what additional information can you provide to us? Mm-hmm. And so that's where we were starting to scratch our heads, because it's true. We did already know that we had a lot of these cases. And I think that's when we started turning a little bit more to the genomic information. How often were we identifying genomes that were completely resistant to all of the antifungals? And perhaps this could be actionable information. That be prepared that if you're starting to test your samples for different antifungals, that you might want to try other therapies if you're not seeing a response from the individuals, from the patients at the hospitals.
Thank you. And then for Dr. Friedman, sounds like a lot of the sampling, the wastewater from the airplanes that you just did were up in Boston, JFK, that kind of thing, as far as folks coming in. Was there any effort to look at once they arrived in the States or at those airports where they went subsequent to that, as far as the manifest details, that sort of thing?
No, so a couple of things. So our wastewater program is in San Francisco, JFK, Boston, Logan, and Dulles. Hopefully it will be expanding through Triturator work at other airports, because the Triturator piece is a lot easier than getting directly from the plane. In terms of, we don't go back and look at who was on the plane or where the travelers, it's all anonymous, even the airlines. I mean, if an airline agrees to have their wastewater taken, they don't want you to tell anyone.
Sure.
There's, competitive marketing issues with one airline versus another. There's liability issues. They're worried that if someone finds out that there's something in the wastewater, they get sick, they're going to try and make a link, even though it would, who knows, you could get infected anywhere. But so we try to be as anonymous as possible.
So I'm sorry, we have about a minute left. So we'll take an online question. We do have a session after this, which is more of a discussion. So go ahead.
All right, so we have a session or a question from one of the breakout rooms, and they're asking for Scott. Can you tell us what kind of industry was discharging those purple and green substances? And also, are you able to tell us, do you communicate this information to your public health departments or labs?
Yeah, so the industry was a dye manufacturer, and they have a pretreatment system that that pretty well takes care of most color that they're discharging. It's not full color removal, but what happened there was a sewer that was breached, and the blue processed, or purple, it's hard to tell what color it was, got into another sewer that was a sanitary only sewer that went to a line that did not go through their pretreatment system. So that ended up not doing what I had shown from the green perspective in our plant, because I think it was a smaller slug. But yeah, so that's kind of how that was. Dye manufacturer, we have a number of those in our footprint. And I think the other part of that question was, do I communicate that to the health departments? I've never seen the need for something like that. Maybe I'm curious as to why we would report something like that to a health department, but I don't know. I work very collaboratively with my local health department. They've heard me tell this story, so they know about it. But prior to that, I don't believe there was any reporting that would have happened on that. But yeah, so that's kind of my take on it. Thanks.
All right. Thank you, everyone. And let's give a round of applause to our presenters today. And I think we're going on a break. Is that correct? A break.
Yeah, thank you again, everybody. We are now on our first break. Oh, just a couple notes. If we didn't get to all the questions that were online, either Slido or Zoom, we'll keep track of those. And again, if you can include the name of the presenter you had the question for, we can get information for you. All right, we're on break. Please return at 10:45 for our next session. Thank you.